you can do that.
And we are back to the presentation. Okay, so first of all, thank you, Paul, for the nice presentation. Very, very nice, and thank you so much for this. And I thank all of you for being here for this seminar, journal club, discussion meeting, and so on. I want to highlight first, this Paul did already, why we are here. So we are involved in this BioTune project, and this is a RISE. And from the acronym RISE, this means it is a research and innovation staff exchange. So I am the first and second bee from Partner 7, University of Camerino, to join Partner 8, UPC Peru. And here you can see on the right, top right, the picture of the kickoff meeting of the that was taking place about 2 years ago now. And so some of us are present here in the picture. So so this is the first secondment, and we expect many more that will take place in the future. Next is about the town. So I'm sure only few of you, except Paul, know where we are. So we are in central Italy on the Apennines, right at the tip of this arrow between Marche region and Umbria region. And this is shown the town of Camerino on its summer dressing, and on the right side on its winter dressing. So it's quite common in January and February to see this kind of picture of the town. What happened in 2016 was a dramatic earthquake. I realized that at any international meeting or presentation outside Camerino, I always talk, before talking about science, I talk about earthquake. But anyway, it was really dramatic for all of us, and so I need to talk about it. What happens is that every 20-25 years, we have an earthquake in the area around Camerino, in the Sibillini area, and the intensity is about between 4 and 5. And every 300 years, so this has been scientifically demonstrated, at least this is what they say, the geologists, we have an earthquake that is of intensity between 6 and 7. And as you know, the Richter scale is a logarithmic scale. So this means there is a huge difference between 4.5 and 6.5. And so what happened was a 6.5 earthquake. And the effects, some of the effects are shown here in these pictures. So on the left, bottom left, you can see a church. This is a church uptown in Camerino, and this is the bell tower that you see only half, because half dropped on this building here. And fortunately, nobody was there, but this is the effect. And on the right side, you can see rescuers immediately the day after the earthquake, working on villages in these Sibillini Mountains. That is a protected area, extremely nice, near Camerino. So these are some of the effects. Another effect was on the rectorate. The rectorate of the University of Camerino was until the 2016 on this very nice building dating back to 15th century, but this has been heavily damaged. So we now have a new campus. So this was constructed 5 years ago. And in addition to this, we have also a lot of work for engineers as well as material scientists. So this is a typical X crack on the wall. And so they analyze all these kind of cracks. So X crack type, L type, Z type, and so on. So that is something specific for material scientists. But for the people of Camerino, this is not so much interesting because most of their buildings are now in this shape. So this is the Bishop building in the main square of Camerino. And as you can see, it is wrapped by this iron or wood beams. So beams everywhere to keep it in place. So this is the effect of the earthquake. Anyway, now we move to Unicam numbers that are quite easy to explain. We have about 8,000 registered students and a quite good professor to student ratio, approximately 1 to 25, and this is quite common for small universities like the University of Cameroon. We have 5 schools or department, whatever is its name. So we work at the Bioscience and Veterinary Medicine school. In addition, we have law, architecture and design, pharmaceutical science, and science and technologies. And within science and technologies, we have math, physics, geology, and computer science. The university has 4 campuses. So the main is in Camerino, as I said, but we have another one in Ascoli Piceno, about 100 kilometers far from Camerino, one in Matelica, about 20 kilometers far from Camerino, and one in San Benedetto del Tronto on the coast, on the Adriatic coast. Then Unicam people. I prefer to talk about people first rather than at the end of the presentation, because I think people should come first. And this is a picture of some of us, picture taken by Anna Maria. And this is Attilio and Maurizio, 2 other researchers in our lab, and Anna and Mara, that you can see here on the right, are 2 technicians, but officially they do research. The pictures are strange, let's say. Anna Maria took this picture, then put on a strange background so that they look like ghost people, but they're real. I can guarantee that they're not ghosts. So coming to the research lines that we are now dealing with, the first is on the translational apparatus, because we have a long-standing tradition and experience on working on protein synthesis. As Paul was mentioning before, our laboratory has been set up by Professor Claudio Gualerzi, and he was working on initiation of protein synthesis and on the translational apparatus. And so he moved all this research that was carried out at the Max Planck Institute in Berlin to Cameroon. So still we're interested in the ribosome, the initiation factor, tRNAs, messenger RNA, and talking with you, I know that you are familiar with this kind of topics and research line. So what we do is that we search for new antibiotics that interfere with translation, and we try to characterize the mechanism of action of these molecules, of these new antibiotics. And recently, this is something that we started a few months ago, we are trying to purify ribosome from pathogenic bacteria to carry out structural and functional studies. In particular, Attilio is working on ribosome from Klebsiella pneumoniae, which is a gram-negative bacteria that is very similar from the morphological and physiological point of view to the model organism E. coli. And so we are trying to purify ribosomes and factors from this pathogen. The second research line is about regulation of gene expression under stress condition. And in this case, we refer to E. coli as a model system. And this is the topic of Anna Maria. And she used to look at genes and proteins that are upregulated and downregulated when the temperature of the E. coli culture goes from classical 37 degrees to 15 degrees or even lower. So something happens, some genes are upregulated, some are downregulated, and this was a field of investigation of Anna Maria. But more recently, she took advantage of the knowledge about a protein that is expressed massively during cold shock. This protein is CSPA, and she used this protein to devise a new method for the detection of in vitro synthesized proteins and peptides. The method is called split GFP, and she has patented this methodology last year, recently, and maybe I will talk to you about this, the details of this split GFP the next meeting or in the next journal meeting. I think it's a nice system, and it is an alternative to the standard and well-known method of using radioactive amino acid. And it is quantitative. The most important thing is that it's quantitative. The third research line is about microplastics. This also, we have been involved since about 1 year, so it's a young research line. As you well know, the microplastic pollute almost any kind of environment: soil, rivers, lakes, sea, of course, they're almost everywhere. And these plastic fragments that are to be considered microplastic, they are usually below 5 millimeter in size, more or less, let's say. We consider that this could be a substrate for the binding and the growth of microorganisms, of bacteria, so they can form biofilm on these fragments. And these microorganisms, they can, of course, they could carry antibiotic-resistant genes. So what we want to investigate is if these particles, these little fragments, can be vehicles for antibiotic-resistant genes in the environment. Next is the focus of this presentation today, and is about the culture collection of microorganisms that we have set up at Unicum, and bio-based products, bio-based chemicals, mainly antibiotics. So what we have done is shown in the next slide. We went outside and we have collected environmental samples from different sources that I will show you in the next slide. And then we bring the sample to the lab, we put on plates, we try to isolate colonies based essentially on morphology using a standard microscope, and we make glycerol stock. And you see here 4 of them that were stored at -80. And so far, for approximately 25–30% of the culture collection that we have done ribotyping. And ribotyping means DNA amplification by PCR of the 16S ribosomal DNA gene, sequencing, and then blast. And from this, we can get an idea about the genus, at least at the genus level. It's more difficult at the species level, but at least at the genus level, we know which kind of bacteria we have. I also have to add an important point that more than 90% of the work has been carried out by Lucia Cimarelli. So another essential part of our laboratory. And she is a really very talented microbiologist. And she managed to isolate most of these strains. And so she is really the one responsible for the CCM, the culture collection of microorganisms. A little bit more about sampling. So we have taken soil samples shown here, river sample or like this, or lake samples, manure, and plant living inside the water, or bacteria that adhere to leaves, plant leaves like this, or that adhere to other kind of substrates like stones or whatever we find in the environment. And last but not least, also this that you can see at the bottom right of the slide, and it is a deep soil sample. So this we did in collaboration with geologists who go into the field, into some fields, and then they do this drilling and they take out pieces of soil to determine the mineral composition, or to establish which kind of foundation they have to do it in case of building constructions and so on. So we call these carrots. And so we break this down and we try to isolate bacteria that may be found at the depth of 5, 10, down to 13 meters. So this is the kind of sampling that we have done. So to summarize what we have in the box, this is shown here, and we have about 1,600 mesophilic strains. We call MES, MES1, MES2, MES3, up to MES 1,600, and also thermophilic strains. So bacteria that have been isolated from, because they were able to grow at a temperature about 60, 65 degrees. And so some of them are shown here, and they are stored at the School of Bioscience and Veterinary Medicine. This is not a picture, but a rendering, so it looks really hot. And we keep it in the freezer, -8. So this is what we have in the box. Before going to describe what we do with this culture collection of microorganisms, I have to mention 3 kinds of consideration. One about the impact we produce, one about the numbers that we have or we may have, and one about the logic of the screen methods. So this will be just 3 brief considerations that may also stimulate some ideas for your work or your future work, because they could be of common application. So first, I said as an impact. So if we consider a cultural evolution of Homo sapiens that is supposed to have appeared something like 200,000 years ago, if we look at the cultural evolution, this parallels some kind of revolution, like a cognitive revolution, the ability to develop a language and communicate, and to prepare and manipulate tools. Another revolution was the agricultural revolution more recently, then scientific revolution and industrial revolution even more recently. So if we look at these stages, different stages of evolution, and we pay attention, we look more carefully at the agricultural revolution, this in principle was based on a phenomenon that is called domestication. Domestication of animals, birds, and plants, maybe also fish, but mainly animals, birds, plants. And this is considered to that was starting something like 15,000 years ago with the dog. So at least they claim that the dog was the first. And this is witnessed by all the paintings, of course. And but what we have to think in connection with our work, what the work we have done, and the microorganism that we have, is that what humans have done it was to put animals in cage, or even if they're not in cage, to exploit animals. In some cases, they have a strong effect on the genetics and evolution of these animals, because they cross, they make all kind of crossing, and so they develop new species. And probably something similar we are doing with our bacteria. So we try to domesticate and put into cages. And in some cases, we do some genetic manipulation, we modify, but for sure we select. We make a selection from those present in the environment, and then some of them we also release into the environment. So the first point is this, which kind of impact we have. So it's difficult to establish, but for sure we have. The second relevant point is about numbers. So we may ask, is it 1,600 big or small? So it's difficult to say, because there are cultural collections of more than 8,000. So from this point of view, it's for sure small. But we have to consider some aspects, like the one that I will show you in the next 2 slides. So first is the work of Satoshi Omura, shown here. And maybe you know that he got his Nobel Prize recently in 2015. And this was based on the work that he did on the discovery and characterization of a drug that is called ivermectin or ivermectin in some other cases, but are the same compound. So what Omura did was to develop a screening for a drug that is active against the nematode. One of these nematodes is responsible for a disease that is called onchocerciasis. So this happens in Central Africa, also in some regions of South America or Central America. And it's terrible. You see here the worm in the eye of a donkey. Of course, it also affects humans and replicates in humans and is responsible for a disease that is called river blindness. And now this drug, these ivermectins that are sold free of charge, this is what they claim, is able to counteract effectively this kind of disease, which is a terrible disease. So this comes from a bacterium, so from a Streptomyces ivermectinus, and this bacterium has been sequenced, and they found that not only ivermectins, so this kind of molecule shown in this oval highlighted here on top left of the slide, but also many other molecules are produced, and there is a total of 37 biosynthetic gene cluster coded into the genome of this Streptomyces. So in principle, there are many more useful molecules there, just in one single organism, in this Streptomyces. So in his words, we should remember, always remember that there is plenty of resources that are hidden in soil, in Japan, everywhere in the world, waiting for the discovery. So this is a very good example of domestication, but also a very good example of the approach that he had. So he managed to develop a screening that was smart to look for and select molecules that were active against the specific nematode he was looking at. In addition to this, so the second example is about the fungus. So this is not a bacterium, it's a fungus, but this Aspergillus terreus that is shown here on the plate, and this is the typical morphology of a fungus like this. Produces lovastatin. Lovastatin and mevastatin are molecules made of a naphthalene like portion of the molecule, a lactone like this, and this part of the molecule that is very good because it makes it soluble. So these are commonly distributed now as drugs because they lower the cholesterol levels in human blood. And so it's, let's say, a very potent drug, and there's also huge business, as you can imagine, around it. But not only this, the same microorganism, so the same Aspergillus, produce this molecule that is a derivative of aconitic acids. So from the anagram, they produce this new name, itaconic acid. Consider that this has the production of this molecule is around 80,000 tons per year. And the reason why is because it is very useful in the plastic chemistry, in the plastic production. For instance, as you can see here on the bottom right of the slide, this is itaconic acid is one that allows the production of shape memory polymers. So polymers that can regain their shape when they are deformed. So again, the same story like the one before for Streptomyces. Aspergillus contains something like 93 predicted clusters. So maybe not all of them are active, but for sure, 2 of them we were smart enough to develop screening to identify this molecule, and then also smart enough to develop technologies to produce these 2 molecules. So in principle, one would need a good microorganism and good screening methods, not so many. In principle, of course. In practice, the story is obviously different. You need many organisms. So this is another good example of domestication. And then last consideration. So as I told you, I am here because of the rice. And the rice is also within the action that are called Maria Sklodowska-Curie action. So this is a picture of Maria Sklodowska. And as you can see, she was not working in a fully equipped laboratory. So she had few things available, for sure, not highly sophisticated equipment or instruments. But she was, for sure, a brilliant researcher. So after all, she managed to get 2 Nobel Prizes, one in physics and one in chemistry. So the same applies for those who were doing bacterial genetics in the 50s, the last century. So one of the most beautiful titles that I found, that I always present in my lecture, is the following: Just Toothpicks and Logic, How Some Labs Succeeded at Solving Complex Problems. It's really surprising in a sense, and wonderful on the other sense, how logic and these simple tools allowed geneticists to identify phages without having instruments to see the phage, lambda phages, and they also managed to identify and discover genetic mechanisms like recombination using simple toothpicks and logic. By the way, and I open here a parenthesis, I find that lambda phage are still could be still a very good model system to investigate, let's say, virus-like system and virus-like genetic elements. But of course, they work only on bacteria. But nobody is working on lambda, I think, since the last 30 years. Okay, so we have these 3 considerations. The impact that we may have during domestication of bacteria. The second is numbers. How many do we need? Probably as many as possible, but 1 would be enough. And the third is a suggestion to, not to you, but to me, first of all, and to all those who are trying to make this kind of work, to use as much as possible logic. Anyway, we have to go into the details of the work that we have done. I'm sure you know that the World Health Organization has promoted this global action plan, and this was on antimicrobial resistance. I know that you are familiar with this kind of topics, so I will not spend time. This is a common phenomenon throughout the world, and bacteria are becoming more and more resistant, and we have less and less drugs that are effective against pathogens. So that is really, really a serious health problem. World Health Organization has defined 12 resistant bacteria that pose the greatest threat to human health. So made a nice table that is very useful to summarize the problem. So we have priority 3, medium, including Streptococcus pneumonia. Priority 2, that is a high priority and contains Staphylococcus aureus, that is the target of the target pathogen that should be investigated in the Biotool project. And critical priority. Among them, Acinetobacter, Pseudomonas aeruginosa, and Enterobacteriaceae, and among them the extended spectrum beta-lactamase producer strains. As I wrote here, is 12 plus 1, because, and you are more expert than me in this, there is an additional critical or supercritical microorganism, that is Mycobacterium tuberculosis, that is very difficult to deal with and very difficult to fight. Okay, so these are the pathogens, and we should do something against these pathogens, focusing on aureus, Staphylococcus aureus, as I said. And so we have these 2 objectives: screening a library of microorganisms, searching for antibiotic producer strains, among pathogens, and in particular Staphylococcus aureus, and produce, purify, and characterize these molecules that are active against aureus, and in particular multi-drug resistant Staphylococcus aureus. So how we did the work? First of all, we need tester strains, and as tester strain we used gram-negative E. coli and Klebsiella pneumoniae. So E. coli is a model system, Klebsiella pneumoniae because it's one of the top priority list of WHO. And we have 2 gram-positive, again a model system, Bacillus subtilis, and the pathogen Staphylococcus aureus. What we did was to apply a very simple, cheap, and relatively fast technique. Again, this has been developed more than 70 years ago. It's very simple. It's called replica plating technique. Maybe some of you are not familiar with this, so I will spend 1 minute to describe what we do. So what is required is a simple and very simple cylinder made of iron and a piece of velvet, sterile velvet. In this case, it's white. This is the one that we have in the lab, and is the real picture is black. But what is important is to have a sterile velvet that you can put on top. Then you have a plate with your tester strain, for instance, Staphylococcus aureus. And what you do, you put the plate upside down on the velvet, and so the velvet is then full of your tester strain, in this case, Staphylococcus aureus. Then on the other side, you prepare 2 organisms of the culture collection, like in this case is MES 1087, and you grow at the center of the plate, and this is MES 946, and you grow at the center of the plate. So you have velvet with aureus on one side, and your organism that you want to test on the other side. So what you do, you press on the velvet, and then you distribute this tester strain. So all these violet ticks are Staphylococcus aureus that may or may not grow in the presence of the strain of the culture collection. So we incubate overnight, well, sometimes even 2 days, and then we look for what comes out. So, and this is what comes out. Sometimes aureus is able to distribute all around the plate, regardless of the presence of these strains. Looks like an egg, after all. And this as well, you see, complete growth all around the plate. In some other cases, you have a behavior like this. This is for Klebsiella, and you see a little zone of inhibition around MES 1531 that I took as an example, or MES 946. You see a slight reduction in growth around MES 946. So this means the bacterium has produced something, and this has diffused in the agar, and this somehow interferes with the good growth of aureus. So very good growth here, very good growth here, slight inhibition here, and slight inhibition here as well. There are, however, cases like this that we have found that MES 79 is able to inhibit significantly Bacillus subtilis. So you see, this is a really strong area where the organism cannot grow. And some of them, like MES 1165, is also able to inhibit significantly. This is more than 1 centimeter inhibition. So imagine the molecules produced by this MES 1165 that then have to diffuse along the agar and around. But this is really strong inhibition. So these molecules are really relevant or of interest for us. So this is an example: no inhibition, slight inhibition, or strong inhibition. This, as I said, is the first and preliminary experiment carried out with the Staphylococcus aureus from a culture collection. So not all the strains of the culture collection have been tested, so approximately 300, and about 10% were active and display, like in this case, a strong inhibition against Staphylococcus aureus. I remark here again, this is an ATCC, comes from the culture collection, so does not carry any antibiotic resistance. So we wanted to move one step forward, and then we went to the next lab, where our colleague, Zemona Petrelli, has a collection of strains that she has isolated from hospital, and so from clinical isolates from patients, and these carry multi-drug resistance. So these are shown on this slide. And you see on top the different antibiotics: penicillin, oxycycline, tetracycline, erythromycin, clindamycin, and so on. And these are strains that are sensitive. You see S all over, sort of a control. And then you start with this SA125, 1 resistant, 132, 2 resistant, 70, 2, 4, 6, 7 resistance, and then 9 resistant. And then you end up with SA19, which carries 10 resistance. So it is resistant to all of them. So these are typical, sort of a gradient of multi-drug resistant Staphylococcus aureus strain. So then we repeated the approach of the experiment, the replica plating with this Staphylococcus aureus. And we got different results. I only show you the best, not because I want to show the best, but just to have an idea of how is the gradient. So the gradient of different microorganisms in terms of activity against the multi-drug resistance can go from 0 to this, that is the top activity. So this MES 1142, as stated on top of the slide, is highly active against all 5 clinical MDR, multi-drug resistant. And as you can see, the halo of inhibition, the zone of inhibition, is quite large. I remind you that S. aureus 19, among all of them, among the multi-drug resistant, carries 10 antibiotic resistant genes. So what we can conclude is molecules produced by and excreted by MES 1142 are able to inhibit also this S. aureus 19, superbug S. aureus 19. Fine and good. Then we ask also the activity, if the activity of MES 1142 can, the same kind of activity is displayed against other pathogens, and that the answer is yes. So this is against Enterococcus fecalis, and this is against Pseudomonas aeruginosa. So this is our additional features of MES 1142. Now I want to talk about another secondment that was the first secondment from the University of San Martin, Buenos Aires, Argentina, to UNICAM. This happened last year, 3 months from April to June 2021. And Alejandra joined our laboratory, so it was a nice experience, at least for all of us. I hope it was also a nice and new approach experienced by Alejandra. She's a biochemist and chemist as background. So we asked her to do something different, to do the microbiologist. And so what she did was to take 50 random strains and test against the Staphylococcus aureus. So these are the results. I show you the best results that she got. Most of the strains were not active, but 3 displayed strong activity, for instance, MES 933, this one, as well as MES 961, this one, against very strong activity against aureus, actually against also E. coli and Klebsiella. And this also gives me the opportunity to show the difference that you can get with this kind of approach. So you can have a growth reduction, like in this case. So there is some kind of slight inhibition around the MES 946, but it's only a growth reduction. So the reduction is impaired. We don't know why, we don't know the mechanism. And this is completely different from inhibition that is shown here. So this can be seen by the naked eye or under the microscope, but there is a strong difference. And of course, as a biologist or like any other biologist, we would be very curious to know which kind of molecule they are and which kind of mechanism they display to show growth inhibition here and only growth reduction here. I also want to add something that is relevant from application point of view. These molecules are as interesting as this molecule because we don't necessarily need to kill and completely inhibit Staphylococcus aureus. Of course, it would be best, but even this kind of molecules can have application, certain applications. So this has been demonstrated, I mean, it has been published. So both molecules are of some interest. So this is the zone of inhibition. And then we come to the last part, that is how to purify these molecules. So it basically consists in 4 steps. First, we grow the bacteria of interest, then we centrifuge down the cells, we remove the cell, we take the supernatant, and we pass through this little column that is a solid phase extraction column. Essentially, it is made of 1 milliliter, more or less, of silica that you can see here, this white, the white region of the column, of the plastic column. So this should be able to trap most of the metabolites, and then these fractions are dried, eluted with acetonitrile, and then dried, resuspended in DMSO, and the fractions are tested on a plate containing aureus to see if we have recovered the activity that was visible in the previous assay that was replicated. So to do this, we use what is called Kirby-Bauer assays. So we take sterile little cellulose filter like this one, and on top of this, we spot 10 microliter of this fraction. And as you can see here, it's possible to recover, in some cases, the activity. And then we pull this fraction and we transfer to HPLC. And again, we applied the same Kirby-Bauer assay, and in this case, activity was still present after HPLC, and in this case, it's concentrated around fraction 59 up to 64, 65. So this is the overall protocol, the overall procedure that we use to purify these compounds. And as you can see, it's really easy to discriminate after HPLC the non-active fractions like this from active fractions. So again, there is a zone of inhibition of the tested strain of the Staphylococcus aureus here. We, as I said at the beginning, have a long tradition in protein synthesis studies. So we were looking for metabolites that can interfere with the protein synthesis. So in this plot is shown inhibition of protein synthesis. So the higher the value, the more inhibited is the synthesis of the polypeptide as a function of purified compound. This is not entirely true because it's not purified, it's semi-purified, so it comes from the solid phase column, but it's enough to discriminate the active, like the red, the green, and the blue, from those that are non-active, like the MES 1142. Remember, that was, I remind you, this was the super active microorganism. So in this case, it is the green line, it is flat. There is no activity against protein synthesis. So this is based on E. coli. So we don't know if the protein synthesis apparatus of Staphylococcus aureus, of course, it's the same. We expect there are some differences, but on this crude assay, it's possible to distinguish something that inhibits protein synthesis from something that does not. There are, like in any procedure, critical issues, and this I want to share with you because it's the other face of the coin and it's part of our work to deal with this critical issue. And these are mainly 4. Sometimes it's difficult to reproduce the production on liquid culture of these metabolites because the production depends on the stage of the growth curve. Usually it occurs on the stationary phase, but one has to select the right window. If you go earlier or later than the right window of production, the yield dramatically decrease. Some of these metabolites do not bind the silica-based solid phase extraction column. So in this case, we have to devise new techniques, for instance, organic solvent extraction to collect this molecule and to concentrate these active molecules. And some secondary metabolites are unstable. So this also happens that they lose activity during the purification steps. So they are active on the plate, they are active after the first column, but then they lose activity after HPLC. Last is that even after 2 steps of HPLC, we find that we found so far that the purification level that we achieve is not enough, is not adequate for mass spectrometry analysis. And this is serious problems we are dealing with and we have to solve. And for instance, in the very first time we have given the products after HPLC purification to a laboratory that is a department of chemistry at Unicam, and they have this sophisticated UHPLC, ultra HPLC, and this is coupled with this Q-TOF liquid chromatography mass spectrometry. So the instrument is very nice and very efficient, very sensitive, but probably for our purposes is too sensitive. So this is just an example of what we obtained. So these are fractions produced by mass 17. Here it is. So this is one of those organisms that produce molecules that are active that as a target, for sure, they're active in the protein synthesis system. And if we analyze fraction 35, we find at least 5 peaks. So this is the list of the molecular weight, but it's difficult to identify which one exactly is the active one. And the same happens with fraction 44, eluted from HPLC. When we analyze by mass spectrometry, we find at least five molecules which are for which we have a very detailed mass, but we don't know which correspond to which in terms of activity. So this is the stage at which we are for some of these molecules, and we should try to go one step further and identify really the molecule that is active in our assay. So to summarize what we have in the box, I told you already, a culture collection of microorganisms. One of the things that could be done with this culture collection is the search for antibiotics that I shown you in this presentation. And we should anyway keep in mind that it is an almost limitless resource, because there may be hundreds or thousands of useful molecules and molecules which are good for biotechnological application in these microorganisms. The midterm plans are to continue the screening for secondary metabolites active against multi-drug resistant bacteria like Staphylococcus aureus, come up with a solution for the multiple peaks detected by MS technique, and characterize the purified molecules which specifically target the translational apparatus. Of course, what I didn't mention is that we not only need the mass and the chemical formula of these molecules, but we also would like to know the structure. And for this, we have to go to large scale preparation of the molecule, and then go to at least NMR spectroscopy lab. But the first step would be to have the exact mass and chemical formula these metabolites. Okay, so I am at the end of the presentation, and of course I have to thank Biotoon, the EU-funded project, but on top of this poll for giving all the Unicam people, and not ghosts, but people, the opportunity to take part in this really nice, interesting, and fantastic enterprise. And of course, I thank all of you for listening.
Thank you a lot, Roberto. It was fantastic. I have so many ideas and questions and annotations here. But let's start first with you guys. If you have any question, raise your hand and you can ask Roberto. The idea now is to ask some questions and then go for some exploration of ideas.
Okay.
If you don't start, I do start. Okay, I give you three seconds.
I see a hand, Victoria Poma.
Victoria, go ahead.
But the audio is off.
Vic.
No levanté mi mano. I'm sorry, I didn't raise my hand.
No?
I would like you to start with the first question.
Sorry, we did not hear you.
That I will suggest that you start with the first questions.
Okay. Vanessa Sarabia, you raise your hand.
Yes, hello, Roberto, very nice
Hello,
presentation. Well, I have it's a very naive question. During the replica plating experiments that you presented, do you see variation of the effect depending on the bacteria? I mean, you presented the example with S audios, but so most of the time this changes depending on the type of bacteria. Sometimes depending on the type of bacteria, you see no effect at all, and then you change, and then you see or more effect or less effect.
Yes. I did not show, let's say, all the possible effects that we see. I only mentioned there is a wide gradient, and this is the real thing, and this is what I expected after all. So there is a gradient in terms of activity, so different bacteria display different production of metabolites against the pathogens. Then there are bacteria that have a very wide spectrum of activity against test strain. I told you, well, I presented in a slide the 4 test strain that are E. coli and Subtilis as a model, and Klebsiella and Aureus as pathogens. And 2 gram-positive and 2 gram-negative. They are, of course, very different. And so we see a very broad range of effects changing microorganisms. Then we also see different effects with purified molecules. So when we take it out, sometime we recover after concentrating on solid phase extraction activity on Aureus, but not on Klebsiella, even if on the preliminary test was positive against both of them. And this probably means that either we lost the activity of one of the compounds, or that the column was able to trap metabolites active against Aureus, but not those active against Klebsiella. So there's a huge amount of work to be done, and develop also techniques that can collect all active metabolites. But as for the replica, yes, we saw really very large gradient of effects. In some cases, halo that are 2 or 3 centimeters. So imagine molecules that diffuse and are still active in 3 centimeters of a plate, far away from the original producer. In some other cases, we find only few millimeters of activity, but still they are, let's say, strong, tough molecules. So we can purify, and we find even after HPLC still active and so on. So it is really a broad range, and it is valid. The statement that I was trying to do in the 3 consideration, or consideration number 2, just one microorganism produce so many useful and interesting things, but sometimes the bottleneck is our ability, our training, or our mind, that we are not able to pick it up, extract, and characterize. But really, the resources are unlimited. So this is what I have experienced this over, let's say, the last 3 years.
Okay, thank you. And sorry.
Into that direction, I managed to observe 2 very interesting halos. One, I think it was 1650 something, where you have a clear plate, like, I don't know, 1 centimeter maybe. And then you have a strong grow nearby the halo, and then it decays again.
Yes. Yes.
Yeah, do you think it is an inhibitor, or it is some sort of communicating molecule that says, Steffi, go away, like swim away or something like that?
Thank you, Paul. This is what a real weird thing. Real weird thing. The answer is still bacteria can do magic things. And they do something on the plate that we have analyzed that it's difficult to interpret for us. So we try to speculate, but it's really difficult. In some cases, they produce, so we see an halo, it's very clear. And then after, let's say, 1 day or 2 days of incubation, then we keep on incubating at the right temperature, and we see some of the Staphylococcus aureus, of the testes, of the pathogens, growing right in the middle of the halo. So not on one border, not on the other, right in the middle. So they form colonies there. Of course, you can think about development of resistance, but in a very short time, and cells that you don't see. So you expect real total inhibition, but it's not. Cells are still there, are sort of quiet, sleeping, and then after 24 hours, they manage to grow even in the presence of these strong metabolites. Are few cells, they form few colonies. So it's not a thick layer of Staphylococcus aureus, are few colonies, but are magic ones.
Yeah, there was also one plate where you had some colonies in the middle of the halo. This sort of resistance, but could it be also some sort of biofilm formation that makes them more tolerant to the compound around?
Of course, because bacteria have many strategies to bypass our stupid assays. So they know how to bypass. Sometimes we know, or we can imagine what they do, sometimes we don't, but they are of highly biological relevance. That is the point. So
definitely,
if we could have time or enough possibility to investigate, they are of highly biological relevance, because they explain some features of these magic microorganisms, but which is the mechanism is mostly unknown.
Yeah, basically you could take one interesting one, you have many of them, and open a new branch of research, which is only developing tests, specific tests for many other molecular mechanisms.
Yes, yes, of course.
For instance, yeah.
Yeah.
Incredible. Yeah, Vanessa Adawi, you have your hand raised.
Yes. I cannot open my video, but it's blocked, I think. Very nice presentation, Roberto.
Thank you.
Yes, I was wondering, now that you exchanged with Paul about these very small colonies surviving the metabolites, the antibiotics, produced by this by your collection, maybe these are persistent cells.
Yeah, of course.
Dormant cells that get tolerant to the drugs and which are very important in the clinic as well, no?
Yes,
Because
right.
they could develop resistance at the end. And then my other question is if you would also address, for instance, latent bacteria, no? Like, this dormant Mycobacterium, no? Tuberculosis that is dormant and that is very refractory to treatment.
Yeah, we didn't test that, of course, but in terms of general problem that you mentioned, like persist and so on, that is true. And what we should remind all of us, I mean, when I talk to my colleagues in Cameroon, we should remind all of us that in textbook, bacteria are just one kind of living form, but they are not. So they have multiple possibilities and they have, let's say, multiple shape, but not in terms of morphology. They have multiple options in the way they live and in the way they display their physiology. So when we approach with an assay like this, like live or dead, killing or non-killing, of course, they can react in different ways. So it's a bit tricky, of course. What I can say, well, we should distinguish the biological problem of relevance. There are many on one side, and on the other side, applications. So applications we desperately need pure molecules, active and temperature resistant, or that they can withstand many steps of the purification. So this is what we need because we want to go further with purification and possibly application. So we have enough bacteria in our culture collection that goes in that direction. They seem tough, resistant, stable, let's say, and we should continue on that. But on the other side, there are many other different conditions that we have seen, and it would really take a lot of time to investigate all of them.
Yes, yes. Thank you.
Yes.
Yeah, and I just continue into that. So you have tested some of them for the translational apparatus, right?
Yes.
So this is something that we have in common, and you mentioned that, okay, this is not the real concentration because maybe we have more molecules than just one in the mixture, and you still need to go one step further to get them pured, or at least to enrich your mixture so you can be more sure by mass spec of what you are having, right?
Yeah.
There I thought of something we did with Attilio long time ago, and I don't know if you have tested it again. So remember when I was working with the luciferases, and there was a reviewer of one of Attilio's papers that asked, but where does it bind? To the 50s or to the 30s? It was a molecule. I don't remember which one. So I think it was 8112 or something like that. And what we did was a very simple assay, which was a pull down with the 30s and the 50s separately, and then tested activity by the in vitro translation of the luciferases that I had set up at the time. Remember, when I was warming the whole room because we could not warm the instruments. To make it to 47 degrees. At that time, it worked very nice. So I thought of something like that. So you have already a mixture of around 10 to 20 components in the same preparation. Why don't you just do a pull down and then do the mass spec afterwards?
Yes, yes, that is right, and I think it's a very good suggestion. After all, yes, I have to say that Attilio had already mentioned that because he has experience, as you said, and we have the possibility to perform this ultracentrifugation in very small volume, very small tubes, and that is fantastic in terms of concentration and efficiency. And I think that that is something that we should do, definitely. Yes, right.
And into the
It could be a very good approach.
Yeah, so just some sort of concentration by target or something. One can give it a fancier name, no? And into that direction also, at one point, so we lack of ultracentrifuges here, and we are not preparing tRNAs for this reason and this kind of thing. So at one point we thought of a method to produce for meal methionine tRNA of method, right? That would not use HPLC and this kind of thing. So the idea was to aminoacylate and formulate the tRNA, right?
Yes.
And then pass it through a column that has immobilized the C-terminal domain of IF2. All right. So
Yes.
you see, you have a mixture of tRNAs passing through.
Yes, yes,
Those ones
yes.
that are formulated.
To trap only the initiator one.
They would, I don't know if they would be totally bound, but for sure they would be slowed down along the flow through, right? And I was thinking on something similar for your enriched preparations. So just imagine that you would have now coli polymerase immobilized. If you would like to find inhibitors of RNA transcription, or maybe directly Staphylococcus enzymes, or DNA gyrases, which are also targets and so on, or polymerases in general, that you may have something like that. So then you pass through your enriched system and then elute your protein, and that goes to mass spec. So you eliminate the fraction of the protein by some treatment. You get only the one that dissolves in acetonitrile and then goes to mass spec probably. Perhaps in that way you can reduce your number of molecules and go directly to the ones that are binding the target. And perhaps this will not take, I mean, it's quite simple in terms of preparing these matrices, right?
Yeah, I think it's a very good suggestion. We should work on this more on this, let's say, analytical technique and analytical methods to trap the active molecules. Yes, yes, this is definitely a good suggestion.
And once you have that and you have the molecular weight by mass spec, then the possible compounds are reduced a lot, and once you know those, you could change your purification strategy to target those more narrowly, and probably you can get higher yields and these kind of things, right?
Yes, right. And when you were mentioning RNA polymerase, now I tell, let's say, the people who are present in this meeting. An assay that is going to develop Anna Maria, I don't know, maybe Paul with your suggestion, is based on molecular beacons. So if you have an oligo with a quencher and a fluorophore, and it has a strong secondary structure, you imagine that there is no fluorescence because the fluorescence is quenched. So if this oligonucleotide has a sequence complementary to a messenger RNA, or yes, complementary to a messenger RNA. So you can set up an in vitro transcription system with a DNA sequence. You take a DNA fragment, RNA polymerase, and then you transcribe. If you produce the messenger RNA, you heat up and then cool down. If you produce the messenger RNA, you should have a strong fluorescent signal. If you produce no messenger RNA, the oligonucleotide would be non-fluorescent. So fluorescent, non-fluorescent. So that was the idea of Anna Maria, if I remember correctly, with some of your hints. And this also is a clever and good system. I mean, we should also investigate that for inhibitors of transcription. And I think this is a good hint also for students. It's a simple assay, not so expensive, even if molecular beacons are relatively expensive, but it's highly sensitive.
Yes, and yeah, definitely. Yeah, but you should tell Ana Maria not to do all the things I tell her to do, because
This, we bought already the beacons, so we are ready to start. She cannot go back.
All right, Miguel, you have a question.
Hi. Hi. Nice talk. Nice presentation.
Thank you.
Well, you commented that some mixtures worked after purifying no longer function. Have you considered the possibility of having inactive enantiomers?
Yes, that is also possible, of course. What we know, I tell you from textbook, of course, is that bacteria are incredibly precise in the production of molecules, in the synthesis of molecules. So they have developed enzymes that, and they can make enantiomers of a very specific kind. But this, of course, does not apply to all molecules and to all bacteria. So, of course, we have to consider the possibility that we have this kind of mixtures. However, we start from something that is, let's say, active in vivo, and we expect that during the purification, the yield of these active compounds should be steady. They should still be available. We guess, but it's just a guess, there is no chemical characterization because we didn't have neither time nor the techniques available. We think that they are somehow out of the cell, they become temperature sensitive. So even at room temperature, somehow they get either oxidized, or even if they are in the MSO, but either oxidized or they are labile. So they lose some of their activity. And that, of course, it's a problem. So we have to change strategy and find either a different approach, or if they are so sensitive, maybe we have to discard this step.
Okay. Well, does your group collaborate with pharmaceutical industry in the field? Maybe, I mean, something like PharmaMar, for example?
No, no,
company that...
Not really.
Okay.
But probably we should, because they have facilities and equipment and know how to deal with this kind of problems.
Okay.
For sure.
Thank you.
I remember Sean O'Connell is his name.
Yeah.
Paola's husband, I think it is, right?
Yes. In Spain.
He's collaborating with a pharma company there. And more than collaborating, they are funding him very often for very small projects to, I don't know, like going to the synchrotron or these kind of things.
Yes.
Perhaps you could talk to him so he tells you more about how that works and so on. Maybe there is a chance either to collaborate with that same pharma, or at least to know more how to deal with them, because probably it would be a nice output to do all this. They have more experience and so on in trying to find and purify these compounds.
I tell you that Attilio has been contacted by Paula and Sean, and they are very, very, very much interested in this ribosome from pathogens, whatever pathogens. They don't have the know-how and the experience for purifying ribosomes, and so that's why Attilio has been involved in this kind of research line. And if we manage to go on with this, then also the molecules could be part of this project. Yes.
Yeah, Kathy is asking, from those 5 isolated mixtures that you have, are they already known as inhibitors? I think you could not know, right?
Yeah, that is another part of the story I did not mention. Let's take the MES17, the last that I showed, and that I showed also the mass spectrum. That is a Penibacillus, and it is also probably Penibacillus polymixa from the 16S ribosomal DNA sequence. From the genome sequence, it's difficult to say. And we know that this produced several antibiotics, among which the famous polymyxin, that is an antibiotic that has been already characterized. So we put on the mass spectra also polymyxin, pure polymyxin, and it has a different elution profile from HPLC, and it has a different molecular weight. So we are on the safe side, it's not polymyxin. But from the mass, we were told from the chemists doing mass spectrometry that these molecular weight do not correspond to what they have in the database. This is good on one side, so probably brand new molecules. On the other side, difficult to guess which is the molecular formula. So you have to work on that. What they say is, we can work doing this. So we have a molecule, let's say 906, and then with the laser we break, and then we get 300 and 600 or so. And from this, we can come up with the kind of type of structure of the molecule, what could be. So that is a typical mass spectrometer approach. And said, okay, fine, do it. And they did it. And so now they also have of each peak, what are the fragments coming from each peak. And that is an additional information. But still, we are missing the link between the activity and the molecular weight of each peak. And that is difficult to achieve because on one side, the instrument is so sophisticated, so sensitive, and requires, I don't know, nanomoles of nothing. And the more you put inside, the more you load, the worse is the quality. So they load very, very little. But the very little amount then is not enough for us to carry on the assay. And so we don't have match between the peak and the activity. So we would need something like 50 runs of UPLC. And after the 50 runs of UPLC, where you can distinguish all the 5 peaks, you take 49 aliquots and you bring to the biological lab and you use 1 aliquot for the mass spec. But so far we found nobody who is willing to do the 50 runs. That is the problem. But that is one possible approach.
And with respect to the databases, because there are many databases out there,
Yeah.
How specialized these guys are in this kind of compounds, or maybe it's a matter of consulting another database.
On
Because
paper,
for-
on paper, everything is easy to do it, and everything is available on paper. We were told, but the real thing, I don't know. I don't know. Nowadays, this kind. Well, I think there are, as for many other things, publicly available database and some other that are kept by pharmaceutical company, private and secret. And that is quite obvious. But for those that are publicly available, should be easy to consult, should not be more difficult than PubMed from my point of view. But I don't know.
you know,
My point of view is
also
not the one of an experienced chemist. So I think in a certain way, I told you, should be as easy as a PubMed, but probably it's not.
But I
And
think
requires also some expertise in consulting these databases.
But I think if you show that experiment where your compound is active against the, the Staphylococcus aureus with 10 resistance, if you show that to a pharma, I think they will just take out their wallet and throw you a bunch of money and say,
I don't want the wallet. I want their databases.
Exactly. Yeah, that's a point that one has to take care. Perhaps with extra steps of purification, target oriented, you may get more homogeneous spectra. And so now keep in mind that also cryo-electron microscopy has become so powerful that they can see magnesium ions, and we saw even a talk where they could see coordinating water in the structures. So this is actually feasible for large complexes like the ribosome or proteins that are above 300 kilodaltons. I'm not sure for smaller proteins how the resolution has gone to cryo. I talk about cryo because the amounts that you need are very small, so that you don't need to go to large scale purifications. And actually you could even work with mixtures. What is the advantage there? That they will see a map, right? A 3D map. Now the question is, can the chemists match their narrowed library of possible compounds that they get from mass spec to such a 3D map? I don't know. So this is, I'm not sure if it has been done or not. Or perhaps a 3D map could help them to sort out which elements or which structure could have, and then design more targeted laser cutting techniques or something like that. Yeah.
Yes, for instance, for what you mentioned before, that are inhibitors that target the protein synthesis, most likely they bind the ribosome. So at the end, the cryo-EM should be able to see those molecules that are ribosome bound and those that are not. And so this is a way to exclude the active from non-active. Yeah, that is a possible approach, of course, in collaboration with some cryo-EM experts. Yes.
Yes. Willie, you had your hand raised. Are you still there? Yes, you are.
Yeah, I was wanted to comment that like there are public databases. Some of them are, I mean, the most popular are the PubChem, Zinc databases, but there are a few databases which are not very known, and they are databases which contains derivatives from natural products. Okay, so there's a database from Brazil, from one of the universities in Brazil. They have derivatives of natural products, and they have plenty of those compounds. Maybe it would be useful. I can just pass the link after the talk about this database. Besides that, you know that pharmaceutical companies, they have proprietary databases, you know, for proteins and for chemical compounds. So instead of, you know, giving you the wallet, they can give you access to those proprietary databases. That could be a goal if in a collaboration with the pharma industry. Okay, that was just my comment.
Yeah, thank you, Willy, and for sure, I appreciate the possibility to have access to the database that you mentioned, and also the comment. It's right. So far, we did not investigate this kind of approach. I mean, contacted possible end user, and for several reasons. One is maybe because we didn't really think of it. But the second reason is that on paper, again, World Health Organization claims that we need a global action plan and we should fight antimicrobial resistance and we should take all possible actions against this, blah, blah, blah. On the other side, in some countries, for instance, Italy, but I think worldwide, pharmaceutical companies have stopped research on antibiotics. So the current status, it is sad to say that people die because of pneumonia, because we have pneumonia-resistant bacteria, and especially old, even not extremely old people, they die of pneumonia. And so it is a real problem. But from the point of view of the pharmaceutical companies, imagine that you have levastatin that I showed, or mevastatin, or these cholesterol inhibitors. So they lower the cholesterol levels, so they decrease significantly the possibility to have heart attack or heart disease. And you start taking this drug when you are around 45, 50 until the end of your life. So imagine the business on that side, and imagine the business for antibiotics that are taken 1 week or 2 weeks at most. So that, at least in the last 20 years, was the strategy of the pharma. Probably something is changing now. We will see after the COVID, there might be a revolution also in these policies. But the overall trend was this. We, and I'm talking about also my former boss, Claudio Gualesti, we were begging at the pharmaceutical company, hey, we have this, we could collaborate, let's do something together. We have the biological approach, you have the money and the interest for application. But the answer was always, no, it's not worthwhile. It doesn't make enough profit for us. So this is a problem, a serious problem.
Yeah, there was a post yesterday of the Wellcome Trust showing this change. This, I think 2 weeks ago, it was communicated that there are more people now dying of MDR than AIDS and malaria worldwide. You know? And then they replied the post, and I asked them, all right, this is something to worry about. How much money is invested for each one?
Quantitatively speaking.
How much money is invested into developing new antimicrobials? I still didn't have any answer, but so my question was into that direction, because lots of money has been invested into cancer research, AIDS, and so on, and not much lately, as you say, has been invested into antibiotics because of the problem of easily reporting resistances. So the last one, I think they took like 3 days to report a resistance after they had been issued for public health. Yeah. So, and this scares a lot also the pharma because they spent a lot of money, and they get a new compound, and after a few weeks they start to report resistances to that new compound, and of course that makes the business unsustainable. The good part is that as you said, there is an increased awareness from national public funding. So you have this XCARP, I think is known, is a public-private initiative just dealing with antibiotics, and with the Wellcome Trust and the NIH, and also now at the EU. So they are including antibiotics within the next potential pandemics, or antibiotic resistance within the category of potential pandemics. So perhaps, let's hope there will be more money for this kind of research, because as we always put on our papers and so on, okay, with the public policies, you can slow down the appearance of resistances or the spread of resistances into the people, but you can only slow it down. You cannot stop it. This is
for sure.
This is impossible. So this is within evolution. So you will always need new antimicrobials. So
yeah,
that's
it's a continuous process. It's not just one step, or so it's sort of a dynamic situation where you provide different, more advanced drugs, and then on the other side, you detect more sophisticated resistance. So it's something that should go on and on.
Yeah, there are many other things that, I mean, you guys have, as you said, the numbers may not look so big, but 1,600 or 1,600 strains are many. So this is this, and as you said, maybe only one bacteria can produce hundreds or thousands of different molecules, and one could spend a research lifetime into just
more than one.
No. And but there is also two things that I have annotated there. So there is also a chance, and this is something we have talked in the lab some time ago, but we have not gone through it yet. And is so you have some bacteria that produces some sort of backbone molecule that then gets modified within the cell, and some of these metabolites get an activity. Okay. And but the backbones are known. Some strains produce a different variant and do other things. So, but the backbone stays there. So until which stage, or what do you think, if we could work with the backbone, all right? And then you have two branches. You have the bioinformatic branch that could be held by Miguel and Willy, for instance, collaborating into creating let's call a virtual library of the derivative of those compounds, right? And then you get computer-based screenings, and then you can get some potential candidates, right? And then the other branch would be a chemist branch, where you do actual combinatorial chemistry using those backbones, and then you get the whole process of isolating the right or the active compounds. Yeah. So this is what we thought: chemical and computer-based. But how much do you know if this is also being done in vivo? So using just one factory, one strain that that you can, let's say, twist to produce a certain family of potential biosynthetic intermediates or other families, and then use your system for screening, because that would be quite fancy, and I probably it's been done somewhere else, but I have no idea. So I'm not updated into that literature.
Yeah, I confess that I really don't know very much, but I've investigated some about these microbial factories. And of course, there are some model organisms that can perform this kind of work. It is again weird because it depends on what you're looking at. And so there's not a single model organism, not a single approach. Looking into literature, there are examples that seems to be very promising, but then I would like to test. I mean, I don't know if they're really trustable or not. One idea was is that there could be also the possibility to produce something like a microbial factory in one organism that can make in vivo this sort of backbone with the possibility to have different branches. So you mentioned branches of approach, and you mentioned the bioinformatic approach and the combinatorial chemistry in vitro. So one is, let's say, virtual, and one is
computer-based.
But what if we could think of sort of a combinatorial chemistry in vivo with the production or overproduction of molecules that have many possibilities in terms of synthesis or branching, in a sense. So, well, that is really hypothesis, but it's something that probably should be investigated. But what you said, I fully agree. It's if we could work on combination of combinatorial chemistry and virtual screening, that for sure will provide so many possible molecules, and then transferring these molecules to a lab where one assay is performed, simple assays like protein synthesis or transcription, that would make everything one step forward. Yeah.
Definitely. Yeah, I think anybody else has some question, comment, final words? Yeah.
So
something like that.
From Miguel. Transcription, no fluorescence detected, and we should do it.
Great. So what I have annotated here, perhaps we can talk about it later. I see a very good possibility for us to continue collaborating. I mean, we're quite good into developing new essays, right? This actually, Katy and Roberto and many of us here like it a lot. what we could start thinking to, thesis, master thesis, or small theses that point into developing very narrow and on-off essays, as you, as Ana Maria is doing now, and Atilio is doing for translation. That on one side. The other thing I have annotated here is, so at one point we have to try these compounds into the essays that we already have, okay? So we have many thread-based essays on translation initiation that are working very nice. So this should be tried with your compounds and mixtures. And also you have seen the data on a totally different essay. That this is just a shot in the dark, but there is this MPROT, so the protease, the principal protease of SARS-CoV-2. And this kind of, so we could also think of setting up these gyrases or DNA polymerases assays where you can pass through a mixture of molecules and capture the enzyme, let it go, and test the activity afterwards directly. Yeah. But to see if something was bound, we should see loss of activity and these kind of things. So very simple and straightforward, that are compatible with small research jobs. And once it is set up here, can be transferred to you guys and use it on your libraries. Things like that. This is what I have annotated. Perhaps at one point we would like to also start a cultural collection here, but we have already talked about it. And first I need to find someone like Lucia, that will stay at least 2 or 3 years taking that seriously and in a very, very, very methodological approach. So this is very important, because otherwise it's just lost effort. And there are some possibilities to find different type of samples. So we always thought of, for instance, natural reserves. We could get access to them. I told you about the boiling river. So we are collaborating with a guy that I totally forgot to invite to your talk. I'm sorry, I told you I would do that, and I just remembered it. But I will tell her that, that perhaps we can meet one of these days. Actually, just freshly doctor from a group in Michigan wrote me last week. She's also working with extremophiles, and she's looking for a postdoc position or a way to come back to Peru or to go somewhere else. So she's just ended her PhD. She will do a short postdoc to summarize some things and get them published. And then she would like to do another postdoc. So she was asking to join my lab, or if I could recommend her somewhere else. She seems pretty fit for this kind of approaches. So we could think also, if we write a project on this, to include her into the plans, to have a trained postdoc either in Italy or Peru or both, I don't know. Yeah, so this is what I have annotated, and yeah, and besides that, I think I'm talking about by everyone. Thank you very much, Roberto.
I thank you to describe a story that I came across while I was preparing this presentation. So without this presentation, probably I would have not Only 2 minutes of your time.
Go ahead.
So once you have a good screening, it's really one step forward and you are in business. However, that is not enough in some cases. So I tell you the story about levastatin or mevastatin that maybe some of you know, or maybe the youngest researchers in this audience do not. So the Japanese are very good screeners. Not only Satoshi Omura, but many others. I found many interesting compounds. So he had devised a screening based on a protein, an HMG, if I remember correctly, that is responsible for controlling the level of cholesterol in the blood. On a murine system, let's say, using the murine model. So he had this specific enzyme, and so he was screening 6,000 strains, probably 6,000 broths, 6,000 different samples. At the end, over more than 2 years, he managed to find one, and this was Aspergillus niger, the one that I showed in the slide. Fine and good. He also managed to identify the molecule. The molecule is mevastatin, and that is active. Fine and good. And then he went to the in vivo system with the mouse to see if that works. That mouse fed with high level of cholesterol, and there was absolutely no effect in vivo. Then he tried with the rat. You know that sometimes mouse and rat behave completely different. I have seen presentation that they have absolutely completely different results, the mouse and the rat. Okay, let's try the rat. The rat, no results. Of course, I guess he was desperate and highly frustrated. I would have been highly frustrated. But, and this is, let's say, a message for you and for all of you, for me and for all of you. He was thinking, but which kind of animal produces a lot of cholesterol? And then the egg contains a lot of cholesterol. So the hen, the chicken, the hen, let's try the hen. And the hen worked. And then the dog worked. And then the monkey worked. And then the human worked. If he would have stopped at the mouse model, he would have thrown in. It's enough, maybe a brilliant idea at breakfast when you're eating an egg, and then you are in business. I find this really not funny, but very useful story, at least for me. So that's why I took 2 minutes of your life.
Definitely. When sometimes we do the opposite. Sometimes one has so many ideas and so on, and if something doesn't work, say, okay, go for the next one. Let's see what happens, and then go for the most likely ones. But perhaps one should stop and have a couple of fried eggs and think, think carefully
with bacon.
what you're doing, and why something didn't work. Yeah. All right.
That's it.
Fantastic, Roberto. Thank you very much.
Thank you again.
Very nice discussion. Yeah, we will update you on the permits for tomorrow, if they allowed us to go or not.
Let's see.
But likely it will be okay. So
fine and good.
Now only Kathy is suffering, poor her.
Poor Kathy.
But the others are still in shape. So we wanted to wait until the end of the day today to see if some other symptoms appear or not. Yeah. So thank you everybody for participating.
Thank you all of you.
See you in the lab.
Paul, we are taking a group photo, or we leave it for?
Yeah, that would be nice if you could turn on your cameras and
yes.
we can take a print screen.
And remember, I don't know who, but someone should add to the slide of this meeting the logo of Biotune and the European Union logo, because that is absolutely required by the coordinator. And then once we have this slide, we send it to the coordinator and we state that this secondment has fulfilled all the requirements, probably. And we also have a video.
Yeah, that's the only thing that they consider important. Not all the important scientific discussions that we had.
But the
and
logo.
everything. No, the logo must be there. So does anybody know how to show only the ones that have the camera on? No.
I
Qué
am
miedo, Miguel.
Sí, Miguel.
Wait a second.
Anna, can you, I don't know how to unblock the video camera.
I think you should. Yeah, you should.
Because I
Marie-
press and I received messages. No se pudo iniciar la cámara de video. Por favor, seleccione otra cámara de video en configuración.
Ah, okay. Okay, so you have to go to the flechita.
Ya,
Start video. This is start video. Hay una flechita, and then you select the camera. You must have the.
¿Dónde, Ana? Perdona. Where is this?
Al lado izquierdo.
Izquierdo. Ah, ya. Ya. ¿Y dónde voy? ¿A configuración? No.
No, donde dice start video o iniciar cámara.
Iniciar cámara. No, no dice eso. A ver.
Donde está el micrófono, Vane. Al lado del micrófono hay una cámara. Al lado de la cámara hay una flechita chiquitita.
Espérame, ¿ah? Qué extraño. Espéreme. Algo ha pasado acá. Ahora sí.
There you are.
Ya, muy bien. ¿Qué tal?
All right. So you can say cheese, whiskey, or whatever you want, but smile. Yeah. 3, 2, 1. I will take a picture. Ready. You can also take some mana because my camera is not so good.
Already did.
Thank you, guys. This will go directly to the European community, so they know that you have been participating. And maybe they will remember when you in the future will apply for funds, and they will say, "Hey, I saw him in
ERC, for instance."
All right.
Okay.
Okay.
Thank you very much.
And bye.
Roberto, I will write to you for having a meeting. An internal
Fine,
meeting.
and I will answer.
Okay, thank you. Through WhatsApp, I will contact you.
Okay, fine.
Thank you all.
Thank you all.
Everybody.
Thank
Take
you.
care.
Bye-bye.
Thank you. Bye-bye. Bye-bye.
Bye.